All right, we are live, yay? I believe so. All right, well, hello, everyone. How are you guys doing? Well, today we're going to talk about stuff and things with Dan Stern Cardinale. Welcome to the show, sir.
Thank you. Happy to be here.
Yeah, we have talked maybe once or twice before, but well, it's glad to have you.
Yeah, once or twice.
Yeah, we talked about having this chat way back. It was like right before. Well, I was in a class. I was in cell biology, and so that class was all over. My whole week was just cell biology, and then Saturday I would just have no cell biology today.
I remember those days. I remember them well.
Oh, do you? Well, we actually had in medical seminar, we had a virologist come and speak to us today because it's like they have a different lecturer every Friday, and so he was a virologist, but he studies the herpes virus.
Though that is a very fun group of viruses. Those are part of the big ones, which are really fun. They do some weird stuff.
Yeah, as I understand, you are a virologist. What is your area of expertise in all this?
So my work in grad school was on the evolution of a really weird, I think somewhat understudied group of viruses, the single-stranded DNA viruses. So viruses are weird because their genomes can be anything. DNA, RNA, single-stranded, double-stranded, they don't care. So I worked with a bacteriophage called Phi-X174. It's a teensy little single-stranded DNA bacteriophage that infects E. coli. And fun fact, it was the first DNA genome ever sequenced.
Nice. Click noise. Yeah, it's just kind of fascinating how the technology for sequencing genomes has just ramped up.
Yep.
It's ridiculous at this point. It's not just one organism a year or just one microbe a year. It's now we're doing multiple consortiums are doing different organisms, nematodes or primates or what have you.
Mm-hmm. And I love the there was somebody in my building, kind of downstairs and around the corner, that was doing a single cell metagenomics. So you just take a single big protist and just extract all the DNA you can and sequence everything. And they were finding intracellular bacteria, they were finding novel viruses, they were finding all kinds of fun stuff in there.
Oh yeah. It's we're in an age now where we can do stuff like that. I love the eDNA stuff. That stuff is really cool in my opinion. Where you just take a sample of lake water or a soil sample or something like that, and then you look at all the organisms that have interacted with this, and they did it for Loch Ness, and Trey the Explainer mentioned it in the video, and it's one of the things they weren't expecting was there were some weirdly large eels living in Loch Ness, but no dinosaurs. No plesiosaurs. Yeah, no dinosaurs.
Sorry for if that. I don't think that's actually. I forget if that's actually a dinosaur, but my apologies if it's not. Yeah, no plesiosaurs.
Oh, it's all right. We'll forgive this one transgression. But
I'm on small things. I'm not up on the big things. Sorry.
Well, small things are very interesting. After all, there is a small thing that has captivated the entire world right now. So
yeah, captivating is one way to put it, because many of us are captives in our own homes at the moment. So yeah, that is an interesting choice of words.
Which is an RNA virus, isn't it?
Yep. Coronaviruses are RNA viruses. They are, I think they're positive sense single-stranded RNA viruses, but don't quote me on that. But yeah, they are RNA viruses, and there are 7 coronaviruses that infect humans. 4 of them cause the common colds, and you get probably 1 or 2 of them every year. And the other 3 are SARS, MERS, and now SARS-2.
Right. Are you saying, sorry, that it wasn't made in a lab? Wasn't manufactured by the Chinese?
surprise, this was definitely not manufactured in a lab. They've done the sequencing on that one, and you find very closely related viruses in bats. And goodness, I wish I had the paper handy, but there's a phylogeny. It's just all the coronaviruses, and you got a whole bunch of bat viruses, and then poof, SARS coronavirus 2 is right in there with the bat viruses. So
yeah,
came from bats.
It's there are some people I've talked to, and they're like, well, how difficult would it be to mutate a virus in this direction? It's like, well, it doesn't appear that way. Certainly you could do it. You could just take inserts from some other organism and like or some other virus, whatever, and introduce them, but it doesn't look that way. It wasn't that any foreign genetic material was introduced. It's just that there were mutations on the existing proteins, which just slightly changed it. Now it's highly contagious.
Yeah, highly contagious and highly pathogenic. Not a fun combination.
Yeah, exactly. And it's kind of amazing how many conspiracy theories have come out of this, which I've just never seen for any other virus that has come into the news.
Yeah, it's you get this confluence of things where people are losing trust in kind of traditional sources of like truth authority on things. So like forget about academia, forget about the media. No one trusts any of that stuff. Or I should say, a significant enough portion of the world just doesn't trust any of that stuff anymore, particularly in the United States. And then you throw onto that the like kind of social media information kind of universe where being kind of sensationalist and provocative is what is promoted. Put all that stuff together and here we are.
Yeah, it's something I think about. Yeah, it's just that I don't remember any other viruses being politicized. Zika, West Nile, Ebola.
Yeah. Well, Ebola. I don't know. Remember the Ebola outbreak? There was that was a big mess. People were up in arms about that, but it wasn't. Nobody was saying it was fake for the most part.
Right. Yeah. That's fair. Yeah. But yeah, this one's weird. This is a new one. Although I guess it's because this is this sort of, is this the first like pandemic of this scale in the information age?
Depends on when you define the information age, I guess, because you had the 2009 swine flu pandemic. The lucky thing about that, to the extent there was a lucky thing, was that it was actually less virulent than your average flu. So that was really lucky. For anyone watching who isn't familiar with the influenza kind of numbers, on average in the United States, there's between 30 and 35,000 deaths attributed to the flu every year. And that fluctuates a lot. On a really good year where you have an effective vaccine and for whatever reason the strain isn't that dangerous and a bunch of people take the vaccine, like the 2009 pandemic, you can get that down to like the 15,000 range. And a really bad year where the maybe the vaccine doesn't work, for example, and that happened a couple of years ago, now you're looking at easily 45,000 deaths, maybe upwards of 50, 60, 70,000. So it's that the 2009 pandemic, fortunately, was not that bad, right? And what we're getting now is something that is approximately as transmissible as that, but significantly more dangerous. So it's an open question whether that would have been, if that had been as dangerous, it's an open question whether that would have been, led to the same kind of social media situation and problems with conspiracy theories and stuff. Interesting counterfactual. Fortunately, we didn't have to find out.
Yeah, conspiracy theories are strange. I was actually introduced to a new conspiracy theory just this week, which is called extinction denialism. Are you familiar with that one?
I had not, but that sounds terrible.
It is absolutely horrible. Yes, it runs in the exact same circle as climate change denialism. So It's like the Venn diagrams of climate change denialism and extinction denialism are the same circle. But that's well, no, we're not going to go political in this one. Anyways, so yeah, there's another one.
We can get political if you want. It's okay.
Well, I did on the channel, on Chesh's channel, when we were discussing this, I got very political. But I think we all know what I'm hinting at. Regardless, or which political party I'm hinting at is behind that. But regardless, Um, so you are in fact a professor, as it happens. Yeah.
I am indeed, yes. My official title is assistant teaching professor. I'm a full-time teacher, which is awesome because I never have to beg for grant money, which makes me very happy.
You don't have to get on your knees before the NSF.
That is correct, which did that in grad school.
Yeah.
Not something I'm jumping to repeat.
I can imagine. Uh, that's really neat. And so, and in your spare time, you moonlight as a creationist debunker.
Yeah, that's my side gig. Is my full-time gig is teaching. I do the general biology course at my school. So Gen Bio 1, Gen Bio 2, fall and spring. So I get like a lot of students every semester. Like every fall, I got like 900 to 950 students. And then in the summer, I do a couple of evolution courses specifically. And since I was in college, I've been really into evolution versus creationism and been kind of engaged in the online conversation of that. And just starting earlier this year, some mutual friends of ours dragged me from Reddit over to YouTube, and so here we are.
And we're very glad they did. Yeah, we've kind of been it our little group of of Reddit YouTube friends keeps growing, which is very nice.
It's great.
Yeah, it's great to network, you know, not just among people, but also across platforms, so that we can get,
Mhm.
you know, as many people as we can all fighting these guys together. And I don't know, it seems like almost there really aren't a whole lot of the opposition left at this point. They're kind of all
There,
congealed into one mass.
there are enough. You know, it's you still have. I mean, you still got the big three, right? You've got CMI, you've got AIG, you've got
Mhm.
create CR or what is it?
ICR.
ICR. Thank you. I always forget the last one. Yeah, ICR. Right. Yeah. It's the third one? EPA. Yeah.
Oh,
So yeah,
yeah,
you
that's
got AIG,
AIG,
CMI,
CMI,
ICR.
ICR, and then the
You've got
Discovery Institute,
Discovery
kind of.
Institute, who is still playing the "we're doing actual science" thing.
Yeah,
We're not really about creation. We're doing real science.
right,
Um, and then you've got the online communities, right? You've got the Reddit community, you've got the YouTube community, you've got a lot of cross pollination between all these platforms, obviously. So,
right,
and yes,
right.
please subscribe to my channel. Oh, it's up
Oh
top. Please
yes.
subscribe to my channel, everybody. It's called Creation Myths, and I basically have two main kind of things that I do there. The first thing is the headline series is Creation Myths, and about once a week I'll publish a video on a creation myth and debunk that myth. And then the second thing is a series I just started last week. We did part two just yesterday,
Mhm.
which is it's a live series called Evolution for Creationists. And what I'm doing is adapting my kind of intro evolution material that I actually use in class. I'm cutting it down into shorter chunks, and we're doing short lessons basically in the basics of evolutionary biology. So we're about to start a three-ish part series on genetics, and I'm really excited. So that's the Evolution for Creationists series. So if you aren't familiar with my stuff, subscribe and come on over. We have fun dunking on really bad creationist science. And I should say it's very specifically focused on the science. I don't get into the theology side of things. It's just like the idea is we're going to take these claims as hard science, and we're going to examine them in that frame and see if they stand up. And the answer is they don't.
Absolutely. Yeah, your videos are fantastic. They are wonderful. They're succinct. They're full of, just packed with information, with relevant technical literature, citations, all sorts of stuff. Your video on the origin of humans in Africa was fantastic. I love that one. Uh,
Thank you.
I It was so much fun because your arguments about how like the well because well uh, the creationist argument, of course, is that humans or you know had a sort of genetic bottleneck in and a founder event in the Middle East, and so they
Right.
spread everywhere else from there.
From
And
there,
mhm,
which makes
yeah,
no sense genetically. You look at the genetic structure of humanity, and nothing is compatible with that. It doesn't work. You can try to
yeah,
fix it, and you can try to fix it, and they come up with like, well, if this lineage was doing this and that lineage was doing that, but then you have other problems with that end up leading to more contradictions. So, like, you could say the African lineage was hypermutating, and that's why it's more diverse than anywhere else in the world. But then that makes the problem with linkage equilibrium worse, because if you had hypermutating lineages in Africa, then you have lots of linkage disequilibrium. But they're the closest to equilibrium, and the rest of the world is at disequilibrium. So you can't even. It's completely like there's no fixing that idea. It's just wrong. And it's wrong for a dozen reasons.
Yeah, I think you went down. Was it 4 different lines of evidence for why this was just?
I went into 4, and I didn't talk about anything. I didn't even talk about like fossils or anything. Like I said, they're fossils. We're going to ignore them, but the fossils tell the story on their own.
Right.
We can ignore that and just look at the genetics. And I ignored. So the 4 that I had, it was the diversity, it was the phylogenetic structure, it was the heterozygosity within the genomes, and then the linkage disequilibrium at the population scale. And I didn't talk about things like alleles that appeared outside of Africa and then ended up back in Africa via migrations kind of back inwards later on. You could follow those around. I didn't talk about anything with like actually documented human history, and looking at things like pictorial records, art, that kind of stuff. I didn't talk about any of that stuff, which is also slam dunk shows that we originated in Africa. There's a dozen reasons creationists are wrong about that, and if I was to go into all of them, it would have been an hour-long video, but nobody wants to watch that.
I don't know. You have a great tone going into these, and so that's why part of why I enjoy listening to your videos is you're very professorly in discussing these things. And you do that
I can't turn it off.
I have to. I read my scripts. I read a script as I'm recording, but you'll just look at the camera and you're like, okay, so we're going to talk about this subject today. We're going to go through these 4 lines of evidence.
Right, and that's really the cross pollination from the teaching, because when I'm in front of a room, I'm walking around. So the only prompts I have for myself is whatever slide is up on screen at the time. I don't even have I don't make any of my things with like presenter tools or anything, because I'm never going to see it. My computer's going to be over there, but I'm going to be walking all over the place. So I got what's in front of me, and I'm basically improvising. And I've had students ask me like, this is a lot of work. How much do you practice each class when before you do it? Practice, like, there's no time for that. You just put it together and you do it. It's and I joke, there's every file that I have, like if I have class at 3:55 in the afternoon, the PowerPoint for that is like last modified 3:53 p.m. It's like I'm tweaking it right up until I do it, and then it's just kind of a song and dance, and that's just how it is.
Right. Yeah. Wow. That is something. I hope to one day. I aspire to have that level of confidence while speaking in front of a group. I took what was it? Public speaking last semester. It didn't really help.
But at the same time, you do this though, which I mean, there's an audience here where you're speaking publicly right now. No script.
Well, I mean, yeah, but it's just the two. There are functionally speaking, there are 4 of us in the room, right? And I can only see you, even though I know the other 2 are there. But but it's like, if I do a talk and it's in front of like 500 people, whatever, on here, I can't see any of them. I'm just doing a debate with this one guy. But I don't know. Something to work on, I guess. But yeah, your videos are fantastic. You cover a lot of very interesting material. I have enjoyed them very much. Do you have?
I appreciate that.
Yeah, your talk on Darwin was fun. I did something like that with Erica on Darwin Day, last Darwin Day.
Nice.
So I guess it's think about like what to do for Darwin Day, because I got to put out a video on Darwin Day. We're evolution people.
Of course, yeah.
That's our like celebrating Darwin Day. It's the one day we get on our knees and pray to Darwin. Yeah. Yep, people subscribe to him. So in your series on teaching evolution to creationists, what are your sort of future plans for that? You said you have a couple of videos on genetics up next. Where do you plan on going from there?
Yeah, so basically what I'm doing is I'm taking there's kind of three places in my over my classes where I teach kind of genetics and evolution. So in Gen Bio we do it, and then in the summer I do the kind of 200 level like intro to evolution course for life science people. And I also do a 100 level course called Evolution, Disease, and Medicine that's designed for non-life science people to like do like a gen ed requirement if they have to do like a natural science. So what I'm doing is I'm kind of taking my materials from all three of those and smushing them together. And so we've done background on Darwin and how he figured out natural selection. We've done kind of the development of evolution since then. And now we're going to start genetics. So the next one, which will be next week sometime, is going to be on Mendel. And it's short. It's just how did he figure out that the old idea of inheritance, the idea of blending, where it's basically like mom and dad each contribute a cell with some fluid in it, and the fluid mixes, and now you get a mix of traits. He showed that was wrong, and we're talking about how he did that experimentally. And then the next one after that will be Mendel's model, the particulate inheritance, and kind of his rules, and very famously Mendel's kind of laws of inheritance, or laws of heredity. And then from there, we're going to do Morgan. We're going to talk about chromosomes and fruit flies. That's going to be awesome.
Oh, you said you like that one.
Oh, yeah. I love that one. And then, because it's the logic of that experiment is just beautiful. And then we're going to, after that, we'll do population genetics. It's going to be like Hardy-Weinberg equilibrium. And then after that, I could go, I got options. And I think what we're going to do after Hardy-Weinberg is go right to selection and drift. Because with Hardy-Weinberg, you could say, okay, you've got this idea where a population is not evolving, and you have to meet these 5 conditions. And each of those 5 conditions is basically like, this mechanism is not happening, that mechanism is not happening, this mechanism is not happening.
Yeah.
So then what I can do is we could introduce the idea of equilibrium and then go through the mechanisms and say, okay, so we could do, genetic drift, natural selection. We'll do definitely do one on like inbreeding and non-random mating. I might push that off to when we talk about like the evolution of sex later on. But we'll eventually cover those 5, mechanisms, mutation and gene flow being the other 2. And from there, then once we do selection and drift, we got options. We could do probably go into like the evolution of novel traits at that point. So we'll talk about stuff like promiscuous proteins and gene duplication and all that fun stuff. And each of these topics is going to take more than one day. So there'll be probably 2, if not 3 days on natural selection, probably 2 days on drift. So, I mean, if I'm doing one of these a week, I've got this planned out for like, I got months to work to work through all this stuff.
Yeah, absolutely. Yeah, the evolution of new traits and the bit about the robustness of and the evolvability of proteins. That's really, in my opinion, very interesting stuff, because the it's not just the creationists who love to go after protein robustness. It's also the intelligent design people, because they have this weird model ish in their head that like proteins are these super fragile things. Like if you have any mutation one way or the other, boom, it's just broken. There's nothing you can do about it.
No, proteins are like that scene in Apollo 13. Now, I'm old, so many of you maybe haven't seen Apollo 13 because that came out in the 90s. But if you go watch Apollo 13, there's a great scene where one of the guys dumps like a bunch of junk onto a table on Earth, and they're like, we got to make this thing fit into this thing using all this junk, because just what they have up in the capsule, and they have to repair like an air filter or something. And that's like how proteins work. It's like, you kind of squeeze it in there and just kind of shove it in, and and it works fine. And that's why you end up with like lots of different ways to accomplish the same thing with different proteins. It's not that you need to have a specific 500 or 1,000 amino acids to get any one thing. You look even among homologous proteins, sometimes you get as low as like 30% amino acid identity, because the only thing that's the same is the active site. And then the rest, the structural part of the protein is, whatever, as long as it stays in place, it's fine.
Yeah. You're right. It's we talked about it in a video, creationist statistics, which we did way back. And that's one of the more popular videos in terms of views. But yeah, we talk about some of the experiments with evolving proteins, artificially with, a certain function into a totally different function. And as far as I can see, creationists and intelligent design proponents really don't ever address this. They don't address any studies on robustness or evolvability. They just kind of have the boilerplate, you can't evolve proteins in a certain time period. It would require this, a certain mutation. It would take this long, and that would be like longer than the universe has existed.
Yeah. So Behe, right? So Behe has that study. I think it was Behe and Snoke from 2005 on evolving a new
2004,
2004 about evolving the new active site in the bacteria.
We actually discussed that in our last video.
Nice. Yeah. So they had a specific target, one. They also had a comically small population size. They only allowed point mutations. They said no selection for the intermediate state. It was like, there was, I think, well, they had different variants where the population size varied across a few orders of magnitude. But there's that famous moment from the Dover trial when Behe was confronted with a paper looking at just how the density of bacteria in soil, and the population that he used for that study, I think it was the biggest population he used for that study, was smaller than the amount of bacteria in 1 ton of soil. And then the lawyer said, and there's a lot more than 1 ton of soil on Earth, isn't there, Dr. Behe? But then you also had, I think more recently, I think it was Douglas Axe from DI had that paper on evolving a specific active site. But again, it was looking at a specific target, not looking at any possible sequence that could accomplish that function. So he's going to find this like 10 to the negative 70th power probability of finding this one specific active site. But like, there's lots of ways to accomplish that same biochemical function. You can't just pick a target and say, because that target is unlikely, you can't evolve that function.
Yeah.
And they just ignore that.
That paper, I think it was it was 1 in 10 to the, I think it was 1 in 10 to the 70th or something like that. It was like a ridiculously tiny number. It's something that's so hilariously small. And you're right. It's the odds of this one sequence, and they just kind of assume that this is like the only sequence that works or that would work.
And they ignore acceptation, so you're not allowed to do one thing and then change jobs. And the problem is, it's not so much that they have these assumptions, that the assumptions are clearly wrong. Because we have examples of where you have a protein that we've observed in modern human history, where you have a protein that does one thing. I'm thinking of a specific example in viruses if people want me to go through the specific one, but like where a protein has one job, and then the virus jumps into a different host and it's constrained in a different way, and that protein acquires a second job. It continues doing the first one, but now you get a completely new active site to do a completely new thing. According to the ID people, that is literally impossible. Like we watched it happen. That happened in HIV, and HIV jumped into humans somewhere in the neighborhood of 1930. So like, it happened in the last century.
Yeah, exactly. It's with the and that's been kind of one of the new avenues of evo devo studies, is how variable proteins are. You have like the modularity of different regions of organisms, and the proteins
I love that stuff. It's really, I'm not well versed in it, but that's a really interesting avenue where you can almost like, I'm thinking of like toys when I was a kid, where you could take like the front end of the helicopter and put the back end of the airplane on it and like put the wheels on the bottom, and you make like some funky machine out of it. Right. Proteins are a lot like that, where you have modular sections that do things. And as long as they don't inhibit each other's roles, you could kind of mix and match and get really weird combinations of functions.
Yeah, it's Colton keeps saying, "Give me a wrench. You're going too fast. I'm trying to give you a wrench." Okay, I think I gave it to him. There we go. All right, Colton, there you go. That's what made arthropods so prolific, is the fact that they are just repeated little modular bits. You've got a segment with legs on it, and you can modify the leg so that maybe it's a leg in these guys, but it's a feeding appendage in these guys, or it's a claw in this guy. And so you can just endlessly modify these things. And that's true.
And it doesn't, and the thing that a lot of creationists misunderstand about this is that there isn't a gene or a pathway that says grow a leg. Right. There's a pathway that says elongate from the midline, branch, right? Fold inwards, right? It's a bunch of very general types of directions. And just by modulating which ones are on and off at different times, you get different structures. You're
No,
not saying
exactly.
build this type of leg versus build this type of mouth part versus build this type of antenna. You're just extending away from the body for a certain amount of time. Then you get a joint. Then it bends back. Like you get just different combinations of signals at different times during development, leading to different structures. But it's the same toolkit. And you could see this. And for anyone who hasn't read it, Neil Shubin's two books, Your Inner Fish and Some Assembly Required,
Both
cannot
fantastic.
recommend them strongly enough. Gets into this stuff. And you can look, for example, at the genes for like finger development or limb development, that you find the same genes in arthropods, that you can take those genes and put them into amphibians or mammals and affect the development in the vertebrates using the arthropod genes, because they're so similar and it's the same underlying pathway.
That's what they did with the eyeless when they moved it from the mouse to fly, wasn't it? Because it doesn't.
Yeah,
It's
that
not
was
saying build them.
that
Go
was
ahead.
the Pack 6, right?
I was it Pax? It might have been, but yeah, it's like
think
they
it
moved
was.
it from the mouse to the fly, and it just says, and it like turns on the pathway to start like the stuff building an eye, and so it's just like you're moving it from from one group to another that's separated by like 600 million years of evolution, you know. uh, it's pretty crazy stuff. But that's how common ancestry works, of
That's how
course.
common ancestry works, and that's how deep homology works. That's the fun part. I really can't, going back a few minutes to my evolution for creationist series, I can't wait to talk about gene histories and convergence and deep homology. That is where it's at.
Oh, let's see. PZ says, or look at bicoid in flies, one of the earliest patterning genes, and it's a fusion protein that has different roles in different lineages. Oh, that's pretty neat. I was
Hello,
reading a paper.
can I just say hello, PZ? I'm just gonna fanboy out for a second here because I've been reading PZ's stuff since like the Dover trial, and been very influential. So hi, PZ.
Yeah, yeah, he's fun. We did also have one from he says hello. We also had a message from CRISPR earlier regarding the Douglas Axe thing. He says Douglas Axe was off by 400 orders of magnitude, the largest error in recorded history. So
That's impressive.
yeah, that's one way to put it. But yeah,
That's
I was reading this paper about the sequencing the genomes of. It's interesting that we're still finding new phyla of like animals, and we're still trying to figure out where exactly they go in relation to other animals. And so they had sequenced the genome of. There's this group of weird little protostomes called mesozoans. It's actually two different phyla, and they thought, and originally back in like
Hey Jackson, I gotta interrupt you for just a second. I really
okay.
apologize. I just have to. For people who don't know, my wife and I had a baby in August, so there's a 6 week old. I just have to go address a small thing
Oh
with the
yeah,
with the
go
small
go
child.
go.
I will be right back, everybody. Talk
Yeah, you're
amongst
good.
yourselves.
You're good. Don't worry about
Sorry.
it. In the meantime, uh, uh, hello everyone. Do we have any? Let's see. Any questions? If you guys have any questions for Dan, put them in the live chat, and we can read those to him if you want. But this is definitely the time when I start juggling. Arthropod eyes got modified into different roles in the insects and arachnid lineages. Yeah, there was a paper on there's this Middle Cambrian sort of, I believe it's a stem chelicerae. It's called Mollisonia, but it's like the antennae were converted into the chelicerae, or the chelicerae were originally used as antennae, and because they were positioned very anteriorly on the head, and they just kind of moved down and are now involved in feeding. So a newbie, yeah, he's got a new little baby. Gunter Wagner's homology book. I have not. The book I'm reading currently is A New History of Life by Peter Ward and Joe Kirschvink, which I have found very interesting. They've said a lot of stuff I had never heard of before. I was just reading today about the Cambrian in their book, and welcome back, Dan.
Hello, I'm back. All right, Mike, we're good.
Yeah, I was just telling them if they have any questions for you, they should put it in the live chat so that we can.
Please do. I would love to hear people's questions.
PZ says, have you read Gunter Wagner's homology book?
Oh, I have not, but I'm putting it on my list.
Yeah, I'll have to write that one down also. Homology, genes, and evolutionary innovation. Thank you, RJ. So, I was talking about the Mesozoans. It's actually two phyla. One is called Orthonectida, one's called Rhombozoa. And what's interesting is, so they've sequenced like one species in each of these phyla, and they've realized they're bilaterians. They're not like stem animals.
Huh.
So they're bilaterians because they have PAX6, even though I don't think they have eyes. But they also have the Spiralian version of Antennapedia. And it's like, why on earth would these guys who look nothing like, they look kind of wormy, but they're not really too much like annelids or mollusks.
But that's weird. I just pulled up the Wikipedia real fast. So it's a Lophotrochozoan.
Yes. Yeah.
Interesting.
They have the Lophotrochon specific version of Antennapedia. But it's like, it makes you wonder, first of all, why do these guys who don't have eyes, why do they have PAX6? Why do they have the bilaterian specific gene? And second of all, why do they have this Lophotrochozoan specific set of genes? If there's no common ancestry between these guys and anything else, why? What's the point?
That makes sense. Yeah. Well, that's kind of gets at for those of you that have had the pleasure of reading Nathaniel Jeanson's Replacing Darwin, he repeatedly makes the argument of, well, creationists, we expect nested hierarchies because you have created kinds and then you have diversification from there. And therefore, evolution and creation explain observations with equal power. We disagree just over how far back you can go doing that, right? But you look at something like this and you see that he's just wrong. You don't have equal power in those two explanations because you wouldn't expect a bunch of these genes that are ancestral and not doing the thing that they do in everybody else in these groups that are these weird little groups that morphologically are kind of on their own, but they have the same genetic toolkit as everybody else, or that's right in their clade. And now creation doesn't have an answer for that, except because that's the way it was made. But that isn't an explanation, right? That's an excuse, right? Whereas evolution is actually a mechanistic explanation for how it got that way. And reading, and yes, for the people in there, yes, the pleasure of that was just on the air quotes, the pleasure of reading Replacing Darwin, the headache of reading Replacing Darwin, and highlighting and taking notes as I went. Jeanson repeatedly says, there's a bunch of these questions where we're equal to each other. We're just as good. And like either he doesn't understand like how scientific explanations work, or I think more likely his target audience is very clearly a non-scientific audience. And he's basically reassuring his audience that, no, it's cool. We have the science too.
Yeah, it's, at the very best, you could sort of, make the nested hierarchy argument for like, what is it, the family level. But beyond that, it just doesn't make sense unless God is purposefully misleading us into thinking that there are hierarchies within hierarchies within hierarchies that are ultimately all unrelated to each other. Mm-hmm. He would have to be, it's like the Omphalos hypothesis or whatever, or just a trickster God to test your faith, whatever. But there's no reason for it above the family level. And so it's absolutely absurd for him to say things like, we predict transitional fossils.
No, they won't call them transitional fossils. They'll call them mosaics because they can't acknowledge that they're actually transitional. So they predict mosaic fossils.
Well, which still though.
And then you have non-expert creationists misinterpreting the word mosaic in that context, because when like people like Jeanson say like mosaic fossil, what they mean is a fossil that has a mixture of traits of different groups, right? But when non scientists, non-specialist creationists, hear or read that, they think it's literally a combination of parts from different
It's a
things.
chimera, right?
Right. They think it's right. They think it's a chimeric fossil where you take the arm from this and the jaw from that and stick it on the skull from this, and you get this weird thing where it's some combination of multiple different
Right.
types of organisms. And that's not what the word mosaic is being used as a replacement for transition, because transition has, evolutionary baggage. There are evolutionary assumptions built into that word, which are not there when you call it a mosaic. But then you call it a mosaic, and then people misinterpret that and they start thinking you take a jawbone and glue it onto a skull, and that's why Homo habilis exists as a species.
Right. Yeah. It's either way. It's heads I win, tails you lose. You just can't bridge the gap because the language is like intentionally loaded.
Yeah. Well, it's intentionally loaded and intentionally, either depending on who's making the argument, either it's just obfuscating, or the people making the argument don't realize there's nuance in there. So you get confusion on what specific words mean, and halfway through a conversation, you go, "Wait a minute, hang on, we're talking about two different things."
A true brain trust.
And I wasn't going to say it, but that makes it hard to have a conversation. When I think somebody in the chat a little while ago pointed out the ongoing confusion between divergence and diversity, which are, for the record,
that was
not
CRISPR,
the same thing.
Yeah. I believe it was Gould's Wonderful Life actually made an interesting distinction between diversity and disparity, and that was a very good book. Have you read?
I have not read Wonderful Life,
Oh,
and I know everyone's like, "What's wrong with you? You gotta." I know. I gotta read Wonderful Life.
It's good. It's not if you follow up on the technical literature, he's not quite accurate about everything anymore. Of course, he wrote it in 1987, I think.
Yeah, so it's going to be a little off.
Yeah. So which is funny because Stephen Meyer basically wrote Darwin's Doubt, which was like a very bad exegesis of Wonderful Life. So 2013 book based trying to squish modern scientific data into a 1987 book. It's like that doesn't really work out so well, does it?
Nope.
Of course, he's not
it's better. Look, you still have. I was just reading a book. It was one of Dembski's books. I don't know why I put myself through this. But it was called Intelligent Design Uncensored, and they spent the whole book. It was short. It was only like 5 or 6 chapters, and 2 or 3 of them were about like the societal and philosophical and moral implications of evolution. But to the extent they talked about arguments at all, they spent the whole first half of the book arguing against Darwinism as though the last 150 years haven't happened.
Yeah.
And I just that's another place where I don't know. I mean, it's one thing to get defend the modern synthesis. Well, it's not 1950, so I don't know what to tell you. But then on the other hand, to have Discovery Institute constantly coming out and railing against Darwinism as though anyone still subscribes to Darwinism, it's
oof. I was reading a conversation that Steve Meyer had with, is it Matthew Perry? I think is his name. He did the Evolution 2.0, which is
Evolution 2.0. Yep.
Which is also just intelligent design, sort of.
Also on my list, I'm working through a long library. I'm getting there.
No, I feel you. I've got lots and lots of books.
By the way, I started Rocks recently, just so you know.
Oh, how do you like it?
It's excellent so far.
Oh, thank you.
I'm sure it will stay that way.
I think so. RJ thinks so. So. But yeah, Myers having this conversation. He keeps referring to neo-Darwinism, and I was kind of thinking in my mind, maybe he's saying the modern synthesis when he says means modern synthesis. Well, he gets to a point where he says in the conversation, well, they've added Mendelian genetics, which they've now added Mendelian genetics, which is why it's called neo-Darwinism. I was like, now added?
What year is it?
It was last year he gave this talk.
We finally incorporated Mendelian genetics into our model.
Thank goodness, right? It was about time. The evolutionists finally caught up with themselves, right?
Been ignoring that guy for over a century.
Yeah, it's wacky. Yeah, referring talking about Darwinism. It's like this term has a meaning. It means phyletic gradualism and natural selection.
Yeah, and it was the kind of unifying idea for mutation and speciation coming out of the 1940s. And like, it's funny, I just shameless plug, just talked about this last night on Evolution for Creationists. We
There
talked
you go.
about the development since Darwin, and yeah, we talked about the modern synthesis, which again, so everyone's clear, it's from the 1940s. It's not what we do now. But this is not
Yeah.
a thing. This is not unique to biology, though, because you still have creationists arguing against uniformitarianism as though the field of geology hasn't gone anywhere since like Lyell.
Yeah.
You know,
Mid 1800s. Yeah.
right? Like again, the field has moved on.
Yeah. These there are just whole fields that creationists do not interact with, and it's not because they don't have the people to do that. I know sometimes some of them try to make the argument like, oh, if we had more people, more funding, whatever, we could. No. You have paleontologists on your side. You have
Mm-hmm.
geologists. You have geneticists. You know, they may be
Yeah,
like, there may only be like 3 of them, but you have them. So you arguing that, oh, we haven't gotten to micropaleontology because we don't have any people is just bullcrap. You do have people. You ignore it because you know it'll screw you. That's why you do it.
yeah, yeah.
And
It's we're gonna focus on these things. We're gonna ignore this other stuff. Um, and you could find like really specific examples of this where Behe is a really nice punching bag on this kind of thing, where you have he's going to talk about like malaria. And one of his books is talking about through his Edge of Evolution. It's like we're going to talk about malaria and specifically about the constraints of malarial evolution. We're also going to talk about HIV and how there haven't been this many changes in HIV. But he's only talking about a little slice of each one, and he's just ignoring this whole field of stuff, which is par for the course for Behe.
sure.
But
Yeah.
there's he's ignoring this whole thing of other evolutionary changes happening in these organisms by choosing to focus on a very narrow slice of them. And there was for people that remember, there was a whole big blogosphere fight over this when over the HIV thing back in the day with one of my favorite proteins, VPU, which is that one I alluded to earlier that evolved a whole new function in HIV compared to SIV that Behe just like never happened, just pretended it never happened. That's how it goes.
Yeah, that's you yeah, I've tried to get if creationists, if they ever talk about genetics, it's some single minuscule thing. Like when I had my the debate with Kevin Anderson, he wanted to focus on like
Yeah.
we're going to talk about this one experiment with fruit flies. We're going to talk about this
We're gonna talk
one.
about. You're gonna talk about what was it? It was it was lactase persistence. He was like, we're gonna talk about this one regulatory scheme.
It's like as though this is representative of all evolution ever. Like this is
Right, because you were taught the debate was on universal common ancestry, wasn't it?
it
And
was.
he wanted to talk about whether lactase persistence represented a loss of regulatory function.
It's like
Which,
we
for the
can
record,
agree to that,
it
but
doesn't.
I mean, you're right. It's like he was wrong because the it was a helicase and not a repressor. But it's like we could even agree that this was a repressor. Okay, fine. Even if we agree to that, the overall
Doesn't matter.
case, it's irrelevant to the overall case. It means nothing.
It's a bank shot of if new traits can't evolve, then you can't get universal common ancestry because you need a bunch of new traits for everything to have evolved from a prokaryotic common ancestor.
Right.
But in making that argument, one, he ignores that like we've observed a bunch of new traits evolving at lots of different scales. And two, he kind of gets lost in the weeds and kind of ends up ignoring the whole point of the debate anyway. And for anyone who hasn't watched that debate, there's a pretty funny moment where Jackson's trying to get Anderson to like figure out what things share common ancestry and what things don't.
Yes.
And he could not get a straight answer, I think, except for dogs and wolves. I think
Dogs?
he said yes. But then it was like any other combination you gave him, you could not get an answer.
Even
Because once you start, and that's by design, because once you start opening the door to like, well, maybe like lions and tigers and some other big carnivorous cats, right? And then once you drop that barrier, then like, but why not this kind of less related group? Why not? I don't what I know hyenas are in there somewhere eventually.
Yeah.
Why not? And the point that exercise illustrates is that when you look at the genomes, there's no line in the sand where you could say, okay, that's the boundary line. It's just the same smooth, decreasing level of similarity going backwards.
Did you see the video that Evograd did on my channel? where he was actually
Maybe.
he did a video. He asked me if he could do a video where he was responding to some videos by Standing and Raw Matt, where they talk about they make this argument that like 60 differences in, was it cytochrome C? No, I don't think that was it. Anyways, they make this claim that like 60 nucleotide differences. I don't remember where exactly it was, but is the demarcation for a kind. And the problem is that
they're using the barcoding argument.
Yeah,
they're saying in barcoding you can actually draw a line.
Yeah, and so it was just hilariously bad.
That makes no sense at all.
You would. It's like if we do this, if you want to use 60, then we group with gorillas, chimps, and orangutans, and like, but you also split the Felidae into a whole bunch of different kinds, and so it just slices the kinds all over the place.
It makes no, and also cytochrome for I mean, unless I have my biochemistry and cell bio completely backwards, cytochrome C oxidase is part of the mitochondrial genome, isn't it? Is that right?
I believe, I believe so. It's involved in the electron transport chain.
I think that's in the I might have that wrong, but I think that's in the mitochondrial genome. Why would you use that? Why not use the nucleus? I mean, obviously there's reasons to use the mitochondrial genome sometimes when you have to make very broad comparisons.
Right,
but
sure.
Like the mitochondria is weird both in its inheritance pattern and also because it's derived from prokaryotes. So things like DNA replication, DNA repair are just different from the nuclear DNA.
Yeah. Yep. You're right.
So it's deliberately picking what you would expect to be an outlier, and it makes no sense.
Yeah, it was hilariously bad, and he just ripped them to shreds over it. Dapper is telling me that I should mention that the inspiration for my most recent video was him. So thank you, Dapper. It's true. You did. I put you in the description of the video because we did a debate. We did a debate with this guy who was a dentist, and he was a creationist, and so Dapper mentioned to this guy how if you take two organisms which are very different today and you go back in their fossil record, you get to a point where they're like nearly identical on both sides of the divide. And he mentioned horses and rhinos, how like the only difference is like their teeth. But yeah, people, if you have any questions for Dan, we will take them because we're not going to be on here very much longer.
Whatever questions you have, by all means.
So questions, people, about viruses or evolution or what shampoo Dan uses, whatever you guys want to know.
Or anti-creationism or whatever.
That too, yeah.
Having a very small child.
Yeah, how is that going?
Oh man. So I've not been doing you mean that I participated in?
Sure.
So I will say that I think the favorite one that I watched was the Dapper Dino like beating his head against the wall to try to get Kent Hovind to understand how we classify dinosaurs. That was hilarious. I think my favorite, honestly, was also like the one that I participated in was also Kent Hovind, because like he's got a script and God bless him, he's sticking to it, and it makes my job really easy and really fun to just be like, thank you for all of that stuff that said nothing about anything that I addressed. So that one was that was an experience. I will say that was an experience.
Oh yeah,
although the one on Neanderthals with Standing for Truth was also a lot of fun. It turned into a bit of a population genetics lesson at one point, but that was a lot of fun.
Well, I mean, when you're dealing with Standing, that's anything's going to become eventually of, okay, let me just explain to you the basics of how we make this argument, and
that's what happened.
Go from there. Yeah, your talk with Hovind was hilarious because he literally said, like, I am moving the goalpost.
He did. He called a goalpost move on himself. Yes. You made him call himself moving the goalpost. Like, yep, that's exactly what you just did. Thank you. Yep.
Yeah, that was hilarious. Dapper says, "Why are viruses such jerks, and why do you hate baby Jesus?"
I don't hate baby Jesus. I think there's a lot of admirable philosophy behind Christianity. I want to say,
It was
I think you could find there's valuable insights in there.
it was just
Some
a
of it
joke.
not, but some of it is. And then
Wire viruses.
wire viruses jerks. Well, they're not trying to be. It's they're the way they make their living is they replicate using a host cell, and when those host cells are us, they often destroy those host cells, and that makes us sick. And then our immune system responds, and those two things interacting tends to make us really sick sometimes, and that sucks for everybody.
Although they are not always, they're also not always jerks because I learned from some assembly required that not the bit about the sing sits, and I already knew that, and we talked about that in our video on coronavirus. The one about the gene that is involved in the formation of memories, which came from a retrovirus.
Retroviruses.
It came from a virus that inserted itself in the ancestor of vertebrates, and then along in the ancestry of tetrapods, we stripped away most of the genes that were associated with it, but we retained one ARC/ARG 3.1, and we use it now for making shuttling proteins, which is involved in the formation of memories, which is really cool, I think, because first I had to look into the literature on that because I didn't know that for our brain to make long-term memories, we have to translate, transcript, or do transcription and translation to make proteins so that we can store these memories. I did not know that, so that was all news to me.
I love that. That was also new to me when I read that book. I love that example. I think it's super cool. And one of the arguments you often find from creationists is that there is not a lot of horizontal gene transfer among eukaryotes. And the more we look, the more we find that is just not true. There's lots of horizontal gene transfer going on in eukaryotic genomes. We just got to know where to look and what to look for.
Yeah, there's some examples among like parasites, right? Like lampreys and salmon and a few other organisms.
Yep. Yeah, parasites in general are under selection to minimize the stuff they take with them, right? So you get things like tapeworms, for example, where they don't have like any kind of digestive system. It's just the skull X to attach, and then body segments that are just gonads, and that's it. All they do is make gametes, and they absorb nutrients through their skin. They don't have anything else because they don't need it.
They even lost their peroxisomes. Cestota have no peroxisomes.
I didn't know that. That's really cool.
They lost a cellular organelle, which I was they just don't need it because I guess whatever, they're sucking on organisms. So I guess you're right. It's just streamlining. It's just whatever we don't. Parasites love to just streamline themselves. So whatever we don't need, just eject it. And so that's how you get things like myxozoans, which are a type of Cnidaria, and they're in the jellyfish group. There's one with no mitochondria.
Was that a recent one they just found? It was It was the first multicellular organism with no mitochondria.
Yep, Hennegaia.
So cool.
I can put it here I can put the name in the. There you go, Hennegaia. Oh, that's misspelled.
It is so cool.
There you go. Yeah, so that's a myxozoan, and there's another. There was another one, another eukaryote. It was a protist. Monocircuminoides was the name of it, and it's a symbiont, so it lives in the gut. There are a bunch of arthropods are kind of strange because a lot of them have like a big protist that sits in their gut, and it has bacteria living all over it, and so they found one that has no mitochondria.
Yes, 3-level relationships. It's very cool.
Oh, can I also ask something? Like I remember also a bit of Orange saying, "I got quote minds." Like it reminded me when you said about Kent Hovind saying, "I am moving the goalpost."
I'm gonna throw the flag on myself.
Oh yeah, I remember that. Well, yeah, I think he thought that. I think that he thought like quote mind meant I just have a quote from like an evolutionist or whatever, but it's like
Nope.
No, hold on, you don't mean what you that's not what you think it means.
Yep, that's not that.
Also I've recently seen a paper where they found a bacteria that is able to synthesize archaean type lipids.
That's interesting.
Yeah. Is it chlamydia? I remember.
I will look it up.
Yeah, I think I remember. There are some bacteria that have like cell membranes that are like part archaeolipids and some part bacteriolipids.
Huh.
Bacteria are weird. They're real
Yes.
weirdos.
Yes, they are.
Anyways, okay. Well, we have been going for just over an hour now. Yep, the membrane lipid divide. We talked about that with, was it Fazal Rana? I think when I had him on. I've actually had him on twice. One time combatively, and the other time we had him on to discuss some biochem stuff, which was kind of neat.
Nice.
He actually he came on and talked about the long-term preservation of soft tissue in dinosaur bones. So that was really cool. You know, not so much a fan of the intelligent design stuff, but that stuff was pretty neat. So at any rate, well, I want to say thank you for coming on. I enjoyed it very much.
I did too. Thank you so much for having me. I really appreciate it. This is great.
Yeah.
I love chatting with you, and it's nice to do it online.
Yeah. If you ever want to come back on, you're always welcome. And I want to say to everyone in the live chat, thank you for watching, everybody. And we will see you next time.
Thank you for being here, everybody.